Good afternoon, everyone. Good afternoon and welcome. It is great to see everybody here today to honor Dr. David Schwartz as the Chancellor's Distinguished Research Lecturer today. I would say we've been thinking about this event for some time, and I'm going to hand things off to the Chancellor here in just a minute, but I would say a few of us were visiting a few years ago. We were talking about all the great research lecture series we've had on campus. We brought in outside speakers, distinguished outside speakers, visiting professors. And I'd say in this very room, I have heard many great speeches over the years. I've learned a lot from the people we have brought in to talk about their science. But one thing that we weren't doing very well is recognizing, honoring, and learning from the research leaders and the scientists in our midst. So with that impetus, this year, we've started the Chancellor's Distinguished Research Lectureship Series. So Donna DeBelia, David Schwartz are the two inaugural holders of that position. And I would say we are extremely honored to be able to, as I said, celebrate and learn a little bit from what Dr. Schwartz has done. Without any further ado, I'll turn things over to the Chancellor.
Thanks, Tom. And this actually should be called the Vice Chancellor for Research Distinguished Lecture Series because it was his idea. Before we begin, I want to recognize two rather distinguished alumni that we have with us today. We have Bruce Schroeffel, who was the CEO of University Hospital for I don't know how many years, but was a wonderful partner for us then. And Dick Krugman is here with us today, who, as many of you know, is one of the longest serving deans of the School of Medicine, I think in the history of medicine, actually. I'm not sure, but in any case, welcome, and we're delighted you're here. We're delighted everybody's here. As Tom said, that this idea is to recognize distinguished researchers from our own faculty, and we're honored to have Dr. David Schwartz with us today to share highlights on what clearly has been an esteemed career of examining how genetic and environmental factors influence the development of complex diseases, including and especially, I think, pulmonary fibrosis. For decades, David has been at the forefront of his field, the last 17 years of which were here at CU Medicine. His leadership roles include distinguished professor, which many of you know is the highest honor that the university gives to a professor of medicine and immunology, associate dean for translational sciences. He was chair of the Department of Medicine from 2011 to 2021, and he previously served as a director of the National Institutes of Environmental Health at the NIH. He's probably happy he's not back there right now. Over the course of an exceptional career, David has made countless contributions to our understanding of the development and progression of pulmonary fibrosis, which, as I think all of you know, is a disease that has limited therapeutic options and no cure. His lab has numerous firsts in advancing knowledge of the disease's origin and possible treatments, which he's working on now. And among them is their discovery of key genetic variants that are critical to the development of the disease itself. His work has made a tremendous impact, and I know we're eager to hear more. But before I do, I want to emphasize to everybody who participates in research on this campus the critical nature of what you do to everything that we are. If you think about it, we have three main missions on this campus. And we all know what they are: research, education, and clinical care. But if you think of that as a kind of virtuous circle, I believe that the keystone of that arch is actually research. It's what draws the best clinicians. It's what gives you the reputation to draw the students. And I think it is, if anything, the single most vital part of our mission. So to all of you who are doing that, my gratitude to you. And with that, I'm going to turn it over to Dr. David Schwartz.
Okay, well, thank you, Don. That was a great introduction, and I agree that our campus here is a pretty special campus, and it's pretty special because of the leadership of our campus. I've been impressed with how our leaders support the shared vision, the shared values of our faculty, and really promote programs that advance our science and also support faculty to take the very best care of our patients. It's a pretty unique place and really well balanced. So congratulations on what you've done here and what the other leaders have done in terms of building an incredible campus and an incredible university. Thank you so much for coming. I mean, looking out, it's just fantastic to see all of you. It kind of feels like I'm either at my bar mitzvah or at my funeral. But thank you for coming. It's a tremendous honor for me to talk with you about my work, and I really appreciate sharing this honor with you. So what I plan to do is to talk about three different questions during this lecture. One, why and how I decided to hunt for the lung fibrosis gene. Second, how this discovery changed my perspective on pulmonary fibrosis. And then what's on the horizon. And I'm going to start first by telling you why I decided to pursue this line of investigation. And there are basically three reasons, none of which had anything to do with being able to be competitive for a grant at the NIH. The first focuses on a patient that I saw 30 years ago. And this fellow was 52 years old. He was extremely short of breath. He had progressive shortness of breath that had worsened over a year's period of time. His chest x-ray was worsening despite multiple courses of antibiotics. And on exam, his exam findings were just localized to his lungs. And he had restrictive lung function and abnormal gas exchange when I tested his physiology. This is what his CT scan looked like. There was a heterogeneous pattern, meaning that he had normal appearing lung adjacent to very severely affected lung. He also had a very peripheral pattern in the distribution of these abnormalities. He had two classic findings for idiopathic pulmonary fibrosis. One, the airways were affected, they were enlarged. And two, he had these honeycomb cysts that were at the base of his lungs. The pathologic specimen that was obtained from him showed, again, heterogeneity, dense fibrosis adjacent to normal appearing alveoli or near normal appearing alveoli. Alveoli are breathing sacs in the lungs. And he also had fibroblastic foci as well as honeycomb cysts, classic features of idiopathic pulmonary fibrosis, and he was diagnosed with idiopathic pulmonary fibrosis. But what was even more impressive about him was his family. So here he is with his brothers, and this is an actual picture of the patient, and you can see that 4 of his brothers died of pulmonary fibrosis, progressive pulmonary fibrosis. A sister died of progressive pulmonary fibrosis. And under my care, he ended up dying from progressive pulmonary fibrosis. So, you know, what struck me was how a family could be wiped out by this disease and what a strong genetic load this suggested to me in terms of the risk of developing pulmonary fibrosis. The second element that pushed me toward this disease is what was known about the pathogenesis of this process. And Jeff Kern, who's here, a friend of mine for the past 35 years now, knows that when we were at the University of Iowa together, this is what the pathogenesis of pulmonary fibrosis looked like. It was a mess. It was everyone's favorite molecule, cell, and pathway. It was sort of, in retrospect, reminds me of the movie Everything, Everywhere, All at Once. It didn't strike true to me that this could be the pathogenesis of the disease. We needed to learn more about it. The third element was that there was a lot to do in this disease. So there were a number of people who developed the disease in the United States and around the world. This disease is increasing in prevalence as our population ages. And the mortality is unacceptably high in individuals who develop this disease. So many of the unmet needs focus on the biomarkers that could be used to identify disease at an early stage, understanding the pathogenesis of the disease, and coming up with new therapeutic approaches. So at that point in time in my career, which was about 30 years ago, I decided I needed to wipe the slate clean and rethink this disease process and figure out an approach that could be completely novel in terms of thinking about what the causes of this disease process were. And I turned to trying to figure out or look into the genetic basis of pulmonary fibrosis. And at that time, there were a few things that were known. Up to 20% of individuals with idiopathic pulmonary fibrosis had a family history of idiopathic pulmonary fibrosis. Complex genetic disorders were associated with pulmonary fibrosis, and workers in laboratory animals clearly demonstrated differential susceptibility to agents that cause lung fibrosis, like asbestos exposures. In terms of thinking about hunting for the fibrosis gene, I had to come up with a method to approach this. And what I decided was to focus on a linkage study. And a linkage study followed by an association study. The linkage done in families with 2 or more cases of the disease, and the association study done in individuals with sporadic pulmonary fibrosis. And there was a strategy behind this. So the strategy was that if we did a linkage study, we weren't tied to anyone's misbeliefs or beliefs about the pathogenesis of the disease. We weren't following a molecule disease or pathway, a molecule or a pathway in terms of the particular hunt for the lung fibrosis gene. In terms of a linkage study, you really need to see a large genetic effect. And I was sure that there was a large genetic effect given the families that had many cases of pulmonary fibrosis among the siblings. And the third element in this was that I knew that I had to go through familial pulmonary fibrosis to understand sporadic pulmonary fibrosis. So I thought that these were connected to each other. And that wasn't a novel thought on my part because BRCA1 had been identified in individuals with breast cancer by studying families with breast cancer. So that was the path that we decided to take back in the late 90s. So the hunt for the fibrosis gene required a lot of people. And Marvin Schwartz here at the University of Colorado, Jim Lloyd at Vanderbilt University, and Mark Steele, who was at Duke University when I first met him and now is here at the University of Colorado, we all came together and we decided we were going to identify families across the United States. And we identified a lot of families with 2 or more cases of pulmonary fibrosis. What happened is we were in a position where we were starting to collect DNA and phenotype the families, but we really didn't know what familial pulmonary fibrosis was. And that's when Mark decided to write a paper on familial pulmonary fibrosis. And basically what he found is that familial pulmonary fibrosis was an autosomal dominant disease with variable penetrance. And that 50% of the families were purely idiopathic pulmonary fibrosis, and 50% of the families were mixed forms of pulmonary fibrosis. And lastly, he showed that this was a gene by environment disease. In other words, he showed that cigarette smoking, in addition to being in a family, was associated with an increased risk of developing pulmonary fibrosis. So this was right up my alley in terms of thinking about complex diseases, gene by environment, and a large genetic load and an important environmental exposure. At about the same time, 2 papers came out. One showing that surfactant protein C mutations were associated with the development of disease in a mother-child pair. Surfactant protein is a protein that's produced in the breathing sacs of the lung, the alveoli of the lung, and they reduce surface tension, so they keep these breathing sacs open. And the second paper showed that familial mutations in telomerase genes were present in 8% of families with familial pulmonary fibrosis. And telomeres are base pair repeats at the end of each chromosome, and here you can see them at the end of each chromosome, that facilitate mitosis, or cells dividing. Not only do cells have to divide, but chromosomes have to divide. And telomeres are essential for that happening. Both of these made a lot of sense in terms of the pathogenesis of the disease. And after spending several years trying to figure out what the genetics was related to pulmonary fibrosis, I thought, well, maybe the genetics had been solved by surfactant protein C and telomerase. Fortunately, at that time, I was working with Marcy Spear, who was a fantastic epidemiologist and geneticist at Duke University. And her perspective on this was that this proved that, that genetics were relevant in terms of the development of lung fibrosis. And this also showed that rare private mutations appear to be specific to unique families, because it wasn't a common telomerase variant across all of these families. It were different telomerase variants across each one of the families. And she said that our approach in terms of approaching this with linkage was really complementary to the candidate gene approaches that these other investigators had used. And then she took a step back and she said, well, what does this really mean for our linkage study? And she said, you know, one of the ways you're going to be able to identify a gene is if you narrow the phenotype. So just focus on families with 2 or more cases of idiopathic pulmonary fibrosis and forget about those other families that have mixed forms of pulmonary fibrosis. And that was really very, very important. So at that time, we went to Decode Genetics, Reykjavik, Iceland, and we worked with Hakan Herkanason and Gunnar Gudmundsson, both friends of ours, who were working at Decode Genetics, and they had a panel of markers, 884 markers across the entire genome. Now, you know what that means? It's just 40 markers on each chromosome. Markers were separated by about 3 million base pairs. So this was a pretty sparse marker panel across the genome, but it was the only marker panel that was available and affordable to us. So we used that marker panel on 100 or so families with 2 or more cases of idiopathic pulmonary fibrosis. And we ended up finding a LOD score. We got really lucky. We found this LOD score of 3.3 on the short arm of chromosome 11. And a LOD score of 3.3 means that there's greater than 1,000 to 1 odds of being linked to pulmonary fibrosis. Quite a finding. We were really, really excited. The problem with it is that the 95% confidence interval encompassed 9 million base pairs because the sparsity of the markers across that region. And it involved over 100 genes, and many of the genes were really interesting pulmonary fibrosis genes. But again, Marcy said, no, don't go after those genes. We're in the interval, and we don't even know what's in the interval. We have to interrogate the entire interval. So we did that. We started mapping the entire interval on chromosome 11 in families with 2 or more cases of pulmonary fibrosis and in individuals with sporadic idiopathic pulmonary fibrosis. And what we found is a significant hit around these gel-forming mucin genes that were relevant to the lung, which was sort of surprising to us, but hadn't occurred to us as being important potentially in terms of the development of pulmonary fibrosis. And so we focused in on this mucin locus on chromosome 11. And the mucin locus was 2 million base pair, from 1.5 to 2 million base pairs in length. It includes MUC5, MUC2, MUC5AC, and MUC5B. And Max Seibold, who was a postdoc in our lab at National Jewish at the time, ended up taking this project on and mapping that region. He's now a full professor at National Jewish and is doing incredibly well. And so what he did is he identified all the markers that were publicly available and developed markers of our own by screening DNA. And he found a region in the promoter of MUC5B that was strongly associated with pulmonary fibrosis, both in families as well as in sporadic cases of pulmonary fibrosis. And what's interesting here, you can see the significance is even more in sporadic than it was in families with pulmonary fibrosis. So just to summarize what I've shown you so far is we've taken families with 2 or more cases of pulmonary fibrosis. We did a linkage study, localized the region to the short arm of chromosome 11, then mapped across that region and identified a promoter variant in MUC5B that was strongly associated with pulmonary fibrosis, both in families as well as sporadic individuals with solo cases of pulmonary fibrosis. And these are the numbers. So 60% of our cohort of individuals with sporadic pulmonary fibrosis had at least 1 copy of the variant. 20% of controls had at least 1 copy of the variant. 1 copy of the variant was associated with an 8-fold excess risk. 2 copies of the variant was associated with a 27-fold excess risk, and it was highly significant. And the world was incredibly skeptical, okay? Because no one had thought that the mucin genes would be important in pulmonary fibrosis. No one had thought that there would be such a strong genetic signal in this complex disease of pulmonary fibrosis. So as a result of that, there were a lot of replication studies. And this is just like maybe 20% of the replication studies that have been done, but they've all shown the same thing. The MUC5B variant is present in 50 to 60% of individuals with disease, about 20% of individuals without disease. 1 copy of the variant is associated with a 5 to 10-fold excess risk. 2 copies of the variant, a 10 to 20-fold excess risk, and these differences are highly significant. In fact, we tested it ourselves because we weren't sure. We sequenced across 750,000 base pairs that included the MUC5B region and lots of DNA before and after the MUC5B gene. And what we found in 3,600 individuals with pulmonary fibrosis and 4,400 controls is the same exact thing. 1 copy of the variant, 5-fold excess risk, 2 copies of the variant, 19-fold excess risk, and you can see this is highly significant, a p-value of 10 to the minus 295th. Now, what I can tell you is, discovery feels pretty good, but confirmation feels better. And I don't think that there's any disagreement that the MUC5B variant is a very strong predictor of the development of lung fibrosis in sporadic and familial disease. We went on to show that this MUC5B variant was equally predictive of early pulmonary fibrosis in the Framingham population, people who didn't know they had pulmonary fibrosis, and that it was equally predictive of the development of pulmonary fibrosis in patients with rheumatoid arthritis. So rheumatoid arthritis-associated interstitial lung disease is a very common form of pulmonary fibrosis, and the MUC5B variant is predictive in both of those cases. Tasha Fingerlin and Anna Pelto went on to show that the genetic landscape of idiopathic pulmonary fibrosis, this complex disease, is actually quite simple, and that the MUC5B variant accounts for 50% of the genetic risk. So they found that there were a handful of rare variants that occur very rarely in the population that are strongly associated with the development of disease. There are a number of common variants that occur commonly in the population that have a minimal risk associated with the development of pulmonary fibrosis. And there's the MUC5B variant that is common in the population and has a very strong risk of being associated with pulmonary fibrosis. This is not dissimilar to Alzheimer's disease. So Alzheimer's disease has a very similar genetic profile. About 30 loci are associated with the risk of developing Alzheimer's disease, and ApoE4 or apolipoprotein E4 is strongly associated. It's a common variant that's strongly associated with the risk of developing Alzheimer's. So what's special about the MUC5B promoter, and what's special about this variant? This variant occurs 3,000 base pairs upstream from the transcription start site. It occurs in a highly regulatory, highly conserved region of the genome. And right away we thought, well, if it's that important, it's got to be associated with gene expression. And Ivana Yang looked at that. Ivana has been a scientific partner of mine for the past 30 years. And what she found is that the MUC5B variant is a gain-of-function variant, really important. She showed that there was over a 20-fold increase in MUC5B expression in the lung tissue if you had the MUC5B variant. And this was true in individuals without disease, as well as individuals with disease, depending on whether you had the MUC5B variant or not. She went on to do an incredibly important study where she asked, well, are there any other variants in that region that are associated with MUC5B expression? And the answer is no. It's actually just the MUC5B promoter variant, 357-05950, that's associated with excess MUC5B expression in the lung tissue, and that's the variant that's associated with genetic risk. So not only is it associated with genetic risk, it is the variant that causes increased gene expression. We were interested in epigenetic marks, and the reason that we were interested in epigenetic marks, and I should say epigenetic marks are really important. They modify what gets expressed in the genome. So some marks will decrease expression, other marks will increase expression. Our MUC5B variant 357-05950 in the promoter of the gene was right in a CPG island. CPG island is where methylation takes place and can affect gene expression. And so the other reason that we were interested in epigenetic marks is epigenetic marks can be influenced by environmental exposures like cigarette smoking. So we knew that some people develop lung fibrosis and don't have the MUC5B variant, but could be affected by epigenetic changes in this region. So we looked first to look at methylation within this region, and we found that methylation around the MUC5B variant was strongly associated with MUC5B expression in lung tissue, the presence of pulmonary fibrosis, and the MUC5B variant. We went on to look at transcription factors, and we found that FOXA2 binds strongly to that region, and that binding of FOXA2 to that region increased the expression of MUC5B. And then Tony Gerber's group, Tony is a professor at National Jewish, Tony Gerber's group showed that this was a hyperchippable site where the DNA opens up and transcription factors promiscuously bind to that region and increase the expression of MUC5B. So this is a highly mutable site that is associated with differential expression that in which epigenetic marks affect differential expression of MUC5B. The next question we asked is, where is MUC5B expressed? And we looked at this histologically first because that's what was available to us, and we stained the lung tissue from about 50 samples of lung tissue. We stained with a MUC5B stain, a MUC5B antibody that stains lung MUC5B positive cells brown. And basically what we found is that MUC5B is overexpressed in what's called the terminal respiratory bronchiole, the airway just before you get to the breathing sacs. And that this overexpression of MUC5B is influenced by the MUC5B variant. Here's an individual with 2 copies of the variant, no copies of the variant. You can see the MUC5B expression is different. And in large population, it was statistically different. Importantly, individuals who are normal do not have expression of MUC5B in this region of the lung. So this is what we would call ectopic expression of MUC5B. It's being expressed in a portion of the lung that it shouldn't be expressed in. We use much more sophisticated approaches to figure out what cells express MUC5B in the lung. So here we used what's called single-cell RNA-seq, which is a technique that analyzes RNA in each one of the cells. So in this plot here, each dot here represents a different cell. Now, what's interesting is you can differentiate the types of cells based on their expression profiles. For instance, these green cells are ciliated cells. These brown cells are alveolar epithelial cells. And what we asked is the question of which one of these epithelial cells are expressing MUC5B. And so what we found is that alveolar epithelial cells, differentiating ciliated cells, goblet cells, and these secretory cells that are seen in the terminal respiratory bronchiole are expressing MUC5B in individuals with idiopathic pulmonary fibrosis. Now contrast that to individuals who don't have pulmonary fibrosis, and you see the expression pattern is dramatically different in individuals with and without lung fibrosis. Importantly, many of the cells that are overexpressed in individuals with idiopathic, that overexpress MUC5B in individuals with pulmonary fibrosis are influenced by the MUC5B variant, and those are highlighted by in these brown boxes. Alveolar type 2 cells and these secretory cells in the terminal respiratory bronchiole. You have the variant, you have more expression. You don't have the variant, you have less expression, even among those with pulmonary fibrosis. So, then we wanted to ask the question, does the concentration of MUC5B affect the development of lung fibrosis? And we did this by creating mice that overexpress MUC5B either in airway cells, and that's the green is overexpression of MUC5B, or in alveolar epithelial cells. And again, overexpression is in green here. And what we found is when we challenged these mice with bleomycin is an agent that we use in the laboratory to cause pulmonary fibrosis. We found that the mice that overexpress MUC5B in the airway and in the alveoli have an accentuated response to bleomycin. They develop more lung fibrosis. And mice that have been knocked out from MUC5B, don't have the MUC5B gene, have less fibrosis when challenged with bleomycin. And these last bars are the bleomycin challenges, and you can see they go down in the mice that are knocked out from MUC5B. This is even better seen when you look at the lung tissue using second harmonic generation. Second harmonic generation is a way of looking at collagen in the lung. And collagen in the lung registers as red here. The green is just background fluorescence. And what we're showing here is that when you have the transgene and you're exposed to bleomycin, you have much more collagen deposition than if you are wild type and you're exposed to the same concentrations of bleomycin. These mice were exposed to very low concentrations of bleomycin that doesn't normally cause very much lung fibrosis in wild type mice. So what I've shown you so far convinced us that MUC5B could serve as a key to understanding many of the unmet needs in idiopathic pulmonary fibrosis. But from my perspective, this was just the end of the beginning of the story, because we wanted to use MUC5B as a way of trying to figure out how we could diagnose the disease earlier, how we could potentially treat this untreatable disease, and also how we could use MUC5B to understand the pathogenesis of pulmonary fibrosis. So I just want to tell you a few of the ongoing studies related to those approaches. So our view is that pulmonary fibrosis takes years to develop. And although established idiopathic pulmonary fibrosis is a rare disease and associated with the MUC5B variant, we found that early forms of pulmonary fibrosis actually are quite common. We looked at the Framingham population, and almost 2% of individuals older than 50 years of age, a lot of people in the audience are older than 50 years of age, have lung fibrosis, and they don't know it. Turns out that the MUC5B variant is strongly associated with the presence of lung fibrosis among those with early disease. So this is a good way of us being able to detect early disease. Also, first-degree relatives of individuals with either familial or sporadic disease have a 25% chance of having pulmonary fibrosis. And they have this preclinical stage of pulmonary fibrosis on their CT scan. This preclinical stage of pulmonary fibrosis is very important to identify because 50% of those individuals progress over a 5-year period of time, and that form of pulmonary fibrosis on the CT scan is associated with an increased risk of mortality over a 5-year period of time, respiratory mortality over a 5-year period of time. Now we have a special population, and our special population are the families that we started collecting over 20 years ago. Now we have over 1,000 families that have been well characterized. We have their DNA in the freezer. We know about their first degree and second degree relatives. We phenotyped many of those individuals, and we have biological specimens on many of those individuals. And those families are important because not only do they have individuals with disease, they have individuals without disease who are at risk of developing disease. So it provides an opportunity for us to focus in on those individuals and try to understand what's causing disease in those individuals, and can we develop the biomarkers to predict who's going to develop disease. And so Joyce Lee and I are leading an effort to identify biomarkers of early disease in the context of genetic and environmental risks. And we just put in a grant this past week to study that problem. So the concept here is that in terms of focusing in on early disease is that the way pulmonary fibrosis is currently treated is you develop disease and then you quickly go into this terminal phase of disease, and you have 3 years to either get a lung transplant or to stabilize your disease process, but it's associated with a 50% mortality once you get into this window. The idea is to diagnose the disease much earlier, increase the size of the therapeutic window, and to use gene-directed therapy to potentially stop the disease from progressing before you develop irreversible lung fibrosis. So let me tell you a little bit about what we're doing to understand the pathogenesis of pulmonary fibrosis. I've told you so far that the MUC5B variant is associated with increased MUC5B expression in the lung, and that expression occurs in the terminal respiratory bronchiole and alveolus, the terminal portion of the lung where the fibrosis actually is. So increased expression where the fibrosis is really helps us connect the dots here. Turns out that if you have the MUC5B variant, you have a 10% chance of developing lung fibrosis. So it has a variable penetrance, just like ApoE4 in Alzheimer's disease. There's a variable penetrance. There are environmental factors, there are behavioral factors, there are probably exercise-related factors that all figure into whether you're going to develop disease or not develop disease. Fortunately, we have a national, international expert in MUC5B who works with us. Chris Evans has his own laboratory here at the University of Colorado. He's in the pulmonary division. He's a full tenured professor, and he does great work in mucin biology. And he's been working with our group for the past decade. And moving from gene expression to excretion of or secretion of mucins into the airway is a complicated process that involves post-translational modifications, glycosylation, packaging, and release into the airway. And Chris is involved in studying all of those processes. And those processes are quite complex. I've talked with you a little bit about the MUC5B promoter and epigenetic marks, but there are other processes that are important in terms of what MUC5B looks like in the cell and when it gets out of the cell. What we've decided to do is to focus on stress in the endoplasmic reticulum. And we decided to focus on stress in the endoplasmic reticulum because ER stress is a common feature of idiopathic pulmonary fibrosis. And ER stress, if it gets bad enough, results in cell death. And there's a lot of cell death in idiopathic pulmonary fibrosis. The other reason we decided to focus on ER stress is that Gang Chen, a collaborator of ours at the University of North Carolina, showed that there was a positive feed forward between ER stress proteins and MUC5B in the distal airway epithelia. And basically, what he showed is that IRE1 beta, an ER stress protein, and XBP1, another ER stress protein, interact, and spliced XBP1 binds to the promoter of MUC5B and causes increased expression of MUC5B. Now, what's really interesting is that increased expression of MUC5B results in more ER stress. More ER stress stimulates IRE1 beta. IRE1 beta splits XBP1, and you get this circular loop. It turns out that when he looked at cells that are polymorphic for MUC5B, have the MUC5B variant, there's increased MUC5B expression and there's increased ER stress, and that increases the feed-forward loop. He also showed that if he knocked out XBP1 with genetic editing of cells, he would decrease the concentration of MUC5B. So this was sort of a direct connection that we wanted to pursue. We pursued this in the single-cell RNA-seq data by looking to see whether MUC5B and XBP1 were co-expressed by epithelia and IPF. I've already shown you the cells that overexpress MUC5B in individuals with idiopathic pulmonary fibrosis. Many of these cells also overexpress XBP1. So the secretory cells especially overexpress XBP1. Really very, very interesting and provocative, makes us think that these two are connected. Now, Ivana Yang and Rachel Blumhagen took the next step. They asked, well, where is it located? Where is that overexpression located? And this is just a histologic specimen from an individual with idiopathic pulmonary fibrosis. And they use spatial transcriptomics to map the cells and map the gene expression of those cells in individuals with idiopathic pulmonary fibrosis. And this is what you get. You find that these cells that are in the respiratory bronchiole are epithelial cells or airway epithelial cells. But what's interesting is when you ask which cells express MUC5B and XBP1, you find that the cells that are in the respiratory bronchiole, the terminal respiratory bronchiole, are the cells that co-express MUC5B and XBP1, suggesting that there's detrimental ER stress in that region of the lung with cell death and involved in the fibroproliferative process. But these are all descriptive. And what we needed to do was to look experimentally what's going on. And we did that in mice. And so we took our mice that overexpress MUC5B and we asked simply, how does that affect the expression of XBP1? And at baseline, you can see that mice that overexpress MUC5B also overexpress XBP1 in terminal airway epithelia. And mice that overexpress MUC5B, when challenged with bleomycin, have a marked increase induction in XBP1 and much more lung fibrosis. We asked the reverse of that. What happens if you overexpress XBP1? And when you overexpress XBP1, you can see that there's increased expression of MUC5B. MUC5B is in black here in the transgenic mice versus the wild type mice. And overexpressing XBP1 when mice are challenged with bleomycin, they have a whole lot of expression of MUC5B, and they develop these cystic lesions in the lung that look like honeycombed lesions. suggesting that the two together are really important in terms of the pathogenesis of this disease process. So what do we think is going on in terms of MUC5B and ER stress as it relates to the cause of pulmonary fibrosis? We don't know. But we know enough to be able to use that to get a program project funded by NHLBI, and that's the focus of our program project. So I'll tell you a little bit about our ideas about how we're approaching this problem. So this is what we would say is the healthy bronchoalveolar epithelium, the airway and the alveolus. You have a bunch of different types of epithelial cells in the airway. You have some progenitor cells that are in this cleft between the airway and the alveolus, and then you have alveolar epithelial cells. Nice, healthy gas exchange unit. Individuals that have the MUC5B variant or for some reason are epigenetically tweaked and increase expression of MUC5B have what we would call ectopic expression of MUC5B, and they have homeostatic ER stress. They don't have cell death. They haven't distorted the architecture of the airway or the alveolus, but they are a vulnerable bronchoalveolar epithelia. Additional injury, we think, results in reprogramming of that bronchoalveolar epithelia. And that results in persistent ER stress, both in the airway as well as the alveolus. It results in apoptosis and cell death, and we know that it results in apoptosis and cell senescence, there are aberrant basaloid cells that begin to populate that area, but never mature beyond aberrant basaloid cells. And we think that there's an arrest of aberrant basaloid cells, and they're unable to repopulate that airway and alveolus following injury because of increased MUC5B expression. There's also activation of fibroblasts, and we think that this ends up accounting for the loss of respiratory bronchioles, which is a cardinal feature of idiopathic pulmonary fibrosis, the development of these cystic honeycombed lesions, and the development of scar tissue. So the conclusions from what I presented are that idiopathic pulmonary fibrosis is a minimally complex genetic disorder, with the MUC5B variant accounting for 50% of the genetic risk, similar to ApoE4 in Alzheimer's disease. Second, that the MUC5B variant results in ectopic expression of MUC5B in bronchoalveolar epithelia. Third, ectopic expression of MUC5B is associated with ER stress and apoptosis or cell death in the right region of the lung that's associated with pulmonary fibrosis. And fourth, MUC5B may be useful in understanding the pathogenesis of lung fibrosis and developing biomarkers as well as therapeutic approaches. So what does this mean for people at risk for the development of pulmonary fibrosis? I think that this type of thinking has really changed and moved the field forward in a very positive way. And it's moved the field forward in a positive way, thinking about clinical and genetic screening for individuals who are at risk, developing intervention strategies and trials to prevent the development of irreversible and progressive lung fibrosis, and the development of novel therapeutics based on gene etiology. I mentioned that there were a number of different genes that are associated with pulmonary fibrosis. So what's on the horizon? What are we really actively engaged in? We're completely focused on trying to understand the mechanisms of MUC5B-induced pulmonary fibrosis. How does MUC5B in the epithelia stimulate fibroblasts to get activated and lay down scar tissue? We don't know that, but we're really working pretty actively with a number of investigators in the program project here and at National Jewish to understand that. Second, we're really interested in this idea of reclassifying pulmonary fibrosis as a molecular classification that's based on genetic, environmental, and clinical subtypes. Because we think that the diagnosis of idiopathic pulmonary fibrosis should no longer be a radiographic or pathologic diagnosis. We think that it needs to invoke the genetics and the molecular biology that we and others are discovering. Third, we think that biomarkers are incredibly important, not only in identifying individuals with early disease, but identifying individuals who develop progressive disease or are more likely to develop progressive disease. Compare what we're doing with idiopathic pulmonary fibrosis to cystic fibrosis. In cystic fibrosis, you have the sweat chloride test that makes the diagnosis of cystic fibrosis and tells you whether someone's responding to therapy or not. We need to think about biomarkers as that type of precision and that type of tool that'll help us identify and take care of patients with pulmonary fibrosis. And the last element is MUC5B directed therapeutics. We need to focus on using MUC5B to biologically and pathologically tweak either cells or animal models or people with lung fibrosis in a way to understand how MUC5B causes lung fibrosis and how reducing MUC5B changes the course of lung fibrosis. So I'm going to close by thanking all of the trainees, the physicians, the scientists, the staff that have worked really closely with us over the past many years, the funding agencies that have been critically important in terms of supporting this work, including our association with industry, and the several institutions that have provided unending support to allow this work to go forward. So thank you very much. I think I'm supposed to ask whether anyone has questions.
And we are recording this session, so if there's a question, I can run the mic up to you. I'm going to go right over here, and then I'll go to Julian next.
David, thank you for an absolutely beautiful lecture. What a wonderful story you had to tell us today. So in the context of the animal models that you described, do you actually have a model of disease that you can use to test therapeutics? And in terms of the pathogenesis, what you've described is, I think, genetic chronic stress, and that typically is associated with increased inflammation, autoimmune or immunological responses. So are any of the therapeutic things that you might model associated with tamping down inflammatory responses?
I didn't hear all of the questions. So are you asking whether we could use our animal model to test therapeutics?
Yes, and in that context, are anti-inflammatories part of the suppression of the pathogenesis of the disease?
Yeah. So the answer to that is a little complex because mice are really different than people in terms of their lungs. Maybe not in terms of other things, but in terms of their lungs, they're very, very different. Mice don't have terminal respiratory bronchioles. They have alveoli that come off of central airways. And so not having terminal respiratory bronchioles makes them a good model for understanding biology. They're not a great model for understanding disease. Now, could they be used to understand biological responses? Yeah, they could be used to understand biological responses. And there is some inflammation that's associated with pulmonary fibrosis. We didn't go into that, but there is a lymphocyte response that's associated with pulmonary fibrosis. We've somewhat discounted that, even though MUC5B is a very important host defense gene and probably results in inflammation in the lung that we're not fully assessing. There are other animal models that are probably better to model lung disease. For instance, in cystic fibrosis, if you knock out CFTR, the cystic fibrosis transmembrane receptor, in mice, they don't develop lung disease. But if you knock it out in ferrets that have terminal respiratory bronchioles, or if you knock it out in pigs that have terminal respiratory bronchioles, they develop cystic fibrosis. So those are the models that we're thinking about. Julie?
Really nice. I was just curious whether you have overexpression of MUC5B in the lungs of individuals that have the telomerase associated mutations. So whether your therapeutics targeting your MUC5B pathway could be used in those cases as well.
Yeah, so MUC5B is overexpressed in all forms of pulmonary fibrosis, regardless of whether or not the MUC5B variant is present. It's expressed more when the MUC5B variant is present. So the idea that we're pursuing is that if it's effective in individuals with the MUC5B variant, it may actually end up being effective in other forms of pulmonary fibrosis, non-MUC5B associated forms of pulmonary fibrosis. Great question. And it's possible that individuals might have the MUC5B variant and the telomerase variant together. Yeah. Oh, sorry.
Hi, David. Great talk.
Thanks.
So my question was almost a follow-up from Craig. And I think these days, most people believe that IPF is also an inflammatory disease. So the question is how the inflammation is connected to the overexpression of the MUC5AB. And because the other thing that you haven't addressed is what happened with the mucin? Do you have any, because normally IPF is not associated to mucus increase, right? But that's the main function of MUC5B. And is it possible that the inflammatory cells, maybe they are just to basically eliminate a tonic production of mucin in the lung, but just
So we know that macrophages take up MUC5B for sure. We don't know what macrophages do when they take up MUC5B and whether that activates macrophages in a way that accelerates the process. We do know that there's a really interesting issue that you're bringing up, which is how could the MUC5B variant occur in 20% of Europeans and only less than 1% of Asians and almost 0% of Africans? How is that possible? And the answer to that is that this MUC5B variant has a positive effect when MUC5B gets overexpressed. And it probably has a positive effect in terms of host defense. When we looked at host defense, or when others looked at host defense in terms of the MUC5B variant in relation to SARS-CoV-2, they found that it was protective, not only in terms of the development of disease, but the extent and severity of disease. So it's really possible that there's a good reason to have this variant when you're young, but maybe not so when you're old. And it clearly relates to inflammation.
Either way.
Yeah.
Great talk, David. I really enjoyed that. So one question I have for you is how much you're thinking a lot about targeting MUC5B downstream and the fact that you get more MUC5B. You've got this variant that's in an enhancer or promoter region. And how much have you investigated what is changed there? Sort of what are the transcriptional alterations or epigenetic alterations that then lead to the upregulation? And could you think about targeting it from that direction too?
Absolutely. We're thinking about that pretty actively. In fact, Tony Gerber has a project on the program project that's focused just on epigenetic regulation of MUC5B. And so the question is, how do you develop a drug really? What's your target? And the approach that our industry partners have is they want to pick a simple target first, which is a small molecule that will decrease the expression of MUC5B. But they want to pick a complex target for the long-term cure of this process, which would be gene editing and trying to understand how and where to gene edit, whether it's the promoter, whether it's an epigenetic mark that would affect expression of MUC5B, whether it's a downstream molecule that's causing the cell damage and the apoptosis.
Hey, David,
and the cheap seats. Great talk. It's a great journey. So it's a bit, you kind of answered the question when you answered it for Mercedes in terms of why would 20% of a population have this? But, you know, it's sort of with the trade-off and sort of with antagonistic pleiotropy because the IPF is affecting older people. But I'm wondering, as a follow-up to that, can you look at differences in pathogens that have been sort of ancestrally present in Europe, Asia, Africa, and so forth? In other words, is this polymorphism perhaps, you know, providing the advantage only for the pathogens that were more prevalent in Europe, perhaps not for the ones that were prevalent in other parts of the world?
So I'll tell you a recent finding that I think is pretty interesting and informative related to that. Anna Pelto looked at the risk of developing lung fibrosis in an Asian population from East Asia. And what she found is that MUC5B is the strongest genetic risk factor for the development of lung fibrosis in East Asians. But among those without disease, the variant is less than 1%. Among those with the disease, the variant is about 3% in terms of minor allele frequency. So about 6% of individuals have at least one copy of the variant. That's a lot lower than we see in Asians. So the question is this due to admixture? In other words, did Europeans mixed with Asians and somehow transmit the MUC5B variant and increase the risk of developing lung fibrosis as a result of that admixture? Anna looked at that really carefully, and the answer is no. It's common ancestral. So it happened before humans migrated out of Africa, that the variant was present. And it probably went under positive selection in Europeans and didn't go under positive selection in either Blacks that stayed in Africa or Asians that moved to Asia. So it's a really interesting idea. And the question is, what are the pathogens? Well, I had a summer student from Princeton once who I gave that project to, and he didn't do anything. So he was a really smart guy. Never showed up at work, gave me a lot of cups that said Princeton on it, but that was about it. It was unfortunate. It's a really good question you're asking. Yeah.
There's been a lot of great questions. Anything else? I would just like to say one thing. I'll pass it to you for a final word. There is a reception following this out in the lobbies. We'd like to invite everybody to join us after that.
Yeah, sure. I'd love to.
Anything else you'd like to say?
Mix with all of you, and thank you so much for this honor. Thank you for taking the time to come.